Recombinant DNA in glow-in-the-dark cats. Recombinant DNA is a fairly new technology that can allow scientists to create neat things that could be beneficial to humankind or could just be entertaining. To understand recombinant DNA, we first need to understand regular DNA. Very basically speaking, DNA is two long strands of variations of 4 types of nucleotides. This sequence of nucleotides codes for proteins, the functional block in living organisms. DNA is first transcribed by RNA polymerase to make mRNA. mRNA is then translated by a ribosome into a chain of amino acids, which, if assembled properly, folds into a functioning protein. You can compare this to a car factory in which computer programs tell robotic arms to assemble parts into whole cars. Because DNA is a long sequence of nucleotides that can be decoded by other organic molecules, some organic molecules can also separate or cut the DNA at a certain sequence of nucleotides. These molecules are called restriction enzymes, and when these restriction enzymes locate a certain sequence which they are programmed to cut, they do so, resulting in 2 DNA fragments. This can be done several times to one original strand of DNA, resulting in fragments of DNA that can be different sizes. These fragments, if cut correctly, can code for useful proteins. This is comparable to isolating a sector of the car factory that manages one specific part. Scientists can implement the structure of DNA in combination with the DNA of the many species of living things around us to create new DNA codes. In the early 1970s, scientists at Stanford and UC San Francisco created recombinant DNA in E. coli bacteria by using a plasmid to put a DNA fragment into the bacterial DNA. Since then, the technology of recombinant DNA has come a long way with scientific advancements, including finding new vectors, such as viruses, to transfer DNA. With new technology, it is now possible to add fragments of DNA to complex organisms, such as goats or cats. One example of the application of recombinant DNA technology is the breeding of goats that produce spider silk. Researchers isolated the gene in the DNA of golden orb weaver spiders, which allows them to create silk, and injected this DNA into goat embryos. Of 7 offspring, 3 showed signs of having silk proteins in their milk. Another example is the creation of cats that glow in the dark. In this case, researchers isolated genes from jellyfish that produce glowing proteins and inserted the genes into cats. The resulting cats can be seen glowing under blue light. As well as being a potentially entertaining application of science, recombinant DNA technology can be implemented in a number of ways to improve current agriculture, medicine, and industry. Recombinant DNA technology can help create plants that are resistant to diseases or drought, new materials, as well as the methods to create them, and cures for many diseases. Adding fragments of DNA to other organisms to enhance their function could revolutionize the future of biological engineering.